Hello, Dr. Lisa. Am I audible?
Good morning.
So, hello everyone. Thank you for joining us at Cajal Week, which is a week-long celebration on the 169th anniversary of Remowa Cajal, organized by Project Encephalon. I am Pratibha Airwal, currently serving as Activity Coordinator at Project Encephalon. I have completed my postgraduate from Neuroscience from the National Brain Research Center. I'm really honored and excited to welcome Dr. Lisa Stowers. Dr. Stowers obtained her PhD from the Harvard University and continued her research on olfactory-mediated behavior with Catherine Dulac. In 2002, she began independent research at the Scripps Research Institute. Her research focuses on how neurons sense and respond to pheromones, especially through the molecular pathways in the mouse brain and which lead to the complex behaviors. I am very happy to be hosting Dr. Lisa Stowers today, and I look forward to listen from her.
Thank you,
Dr. Stowers.
Thank you, Prahibna. Thank you, Tanya, for the invitation, and thank you to the organizers. This is a really lovely event. Let's see. Can you see my screen? Not yet. Sorry. Let's try again. Sorry.
Yeah, excuse me.
There we go. Is that fine?
Yeah.
Okay, great. Thank you everybody for joining us. It's early morning here in California where I'm at in the United States, but I know everybody's at different times and I really appreciate you staying to the end of the day. My lab is interested in understanding the neural mechanisms that underlie social behavior. Social behavior is a really great model to study the brain because it relies on all the computations that other labs focus on independently, such as learning and memory, sensory integration, motor output, decision-making. And so we use that as a model to look for general features and principles in neural coding. And one of our focus is using the transformation from sensory information into behavior. And often we sort of treat it as something that if you just flash it, if you change the environment, everyone's brain will respond to it in the same way. But as we know from our own experience or even working with our model organisms, that's not the case. Every individual responds to the environment slightly differently. And that depends on their past experience, their developmental state, or even their internal state, whether they're hungry or stressed or tired. And so several years ago, my lab started focusing on this internal state modulation more closely. And the question that we started with was a simple question that was easy to study in mice, which is the organism that my lab uses. And we look at the effect of the olfactory system on behavior. And it's common knowledge, and we all understand that male cues attract females, but it doesn't always attract females. It depends on her receptive state. And so it depends on her ovulation cycle and whether she's ready to mate or not. And in the mouse, we call this estrus. And the estrus cycle in the mouse is very short in our lab animals. So from estrus, where she's receptive and interested in mating with males, until diestrus, where she is refractive and will not mate with males, it's only about 2.5 days. The whole cycle takes 5 days. So every 5 days, she's going through these phases of being receptive or attractive. And the odor cues of males, we can look at the behavior of the female. So here we take male odor cues and place them on one side of a cage and have a female in the cage. And the blue lines are the tracks of where she's moved throughout this 10-minute experiment with the control odor on the other side. And the warmer colors indicate that she spent longer time in that area. So you can see when she's in estrus in this top figure here, she's spending more time near these male cues. She's more interested in them. But during diestrus, she's less interested, perhaps even a little aversive. And so with this robust assay in mind, over the years, we had identified several cues that were present in the mouse odor. And one of them, shown by these gray blobs here, is an attractive cue. And it's this one element that the female is attracted to. And during diestrus, she's just not really paying attention to that. And we wish to understand how the brain changes in this short period of time to alter her behavior. But first, the postdoc in my lab, Sandeepa Day, asked a really important question. And she wanted to first control and make sure that it wasn't the nose that was changing, it was the brain that was changing, not the perception, but the response to it. And I thought this was not a very useful question, because we all know that the sensory organs are there to gather as much information from the outside world as possible so that the brain has a chance to actually act on it. And so it was common understanding that the nose would not change and that it would be something about the hormonal action on the brain that would change. And we know this to be the case just from back pre-COVID, when we used to be in crowded rooms very easily. And you can imagine in this situation, there's very loud noise everywhere, but you can have a conversation with someone just by focus and attention. And all the other noise in the room sort of recedes into a din. You don't really hear it too much. You can really hear the person you're talking to. But if somebody across the room says your name, your brain will hear it, know the direction it came from, and even if you're familiar with the people, know who the speaker was. So that meant all the other information that you weren't hearing while you were having that conversation was getting into the brain, and the brain was deciding how to act on it. So it's based on this phenomenon and many others that we assume that the brain is the thing that's changing during estrus and diestrus. But the experiment was very easy. And Sandeepa noticed that estrogen is the female hormone that surges during estrus when she's responsive, and progesterone is the hormone that surges when she's non-responsive. And she could just remove these hormones and start with a basal level of neurons in the mouse by giving her surgery to remove the ovaries, which secrete these hormones. And then just ex vivo in a dish, look at by calcium imaging at the neuronal activation of these neurons during estrus and as she added back physiological concentrations of the hormone. And so this was a really simple experiment, just took a week or so to do, so might as well give it a go. And what happened was actually extraordinary to me. So these are the traces of the calcium imaging from these neurons. So these are 4 or 5 neurons. And when she perfuses on the pheromone, the male odor, you can see these neurons are responding to it. They'll do it twice. And then when she adds not estrogen, but the physiological concentrations of progesterone, and waits just a short period of time and it perfuses on the odor, the pheromone, the neurons do not respond at all. Now these are in a dish, they're ex vivo, so to make sure that they weren't just dying, we added an antagonist of the channel itself, and you can see all the cells are perfectly alive, they're able to respond. So something interesting was going on here. And I really like this data because this is suggesting that the sensory neurons themselves are sensitive to the progesterone, because these neurons are ex vivo, they're separated from the brain. So it's not the brain that's telling these neurons, you know, shh, don't respond, she's in diestrus. The sensory neurons in the nose themselves are knowing it. And it's also happening quite rapidly. So sex steroid hormones are known to act as transcription factors, and they usually take about 90 minutes to evoke transcription and change the activity of neurons. And here we're getting a very rapid action phenomenon that's happening cell autonomously. So because it was so unexpected for me, we went and Sandeepa found the mechanism. Excuse me. And this is what's happening. So this is the textbook example of what we knew to be the signaling in the olfactory neurons. That's a specialized olfactory neuron in the vomeronasal organ of the mouse. Excuse me. And here we have G protein couples receptors that are specific for the type of ligand that it's binding. So in this case, this one's responding to the male urine odor. And then it's going through G proteins, through phospholipase to gate this TRP channel. And that's what's happening here and here. But Sandeepa also found that these neurons express a non-canonical progesterone receptor. And when it binds progesterone, when she's in diestrus, it starts this kinase cascade that specifically targets this enzyme, the phospholipase, and silences it completely. So no matter how much pheromone is out here, these cells, excuse me, will not respond. And that's what's happening here. So it's this rapid action signaling mechanism that is silencing the brain. And what's in effect, what's happening is when the female is in estrus and she smells the male odor, all the neurons, including the male odor, are able to activate the sensory neurons. And the brain says, yay, there's a male out there. But during diestrus, a few days later, when the progesterone silences the cascade, she can smell the other cues, but not that attractive cue. So she has no behavioral response to it. It's a complete sensory gate that blocks the behavior entirely. If she can't sense it, she can't respond to it. So over the years, this work was published several years ago, and we've been thinking a lot and have been challenged by why this would occur. Because again, I believe that the job of the sensory system is to gather as much information as possible, and that the brain should be the one to act if progesterone do not respond. That should be the calculation the brain makes. Instead, this is happening in the nose. And so folks have told me that perhaps the poor female's brain is just too limited to be able to respond to male cues when she's in diestrus, when she can't mate anyhow. So don't distract her. Don't give her that extra sensory information. That evolution has evolved to keep things simple in the female brain. And I think that's kind of silly, because we're bombarded with tons of sensory information every day. And it's the idea of actual sensory overload, especially to something so important as a social cue, is not really demonstratively important. There are some certain types of mouse behaviors where the female cannot reproduce if she smells another male. So if she just was impregnated by a stud male and he runs off and a strange male comes in her place, she will spontaneously stop the pregnancy so she can mate with the new male, so that she'll have a parental, a family group for the young animals. And so perhaps this is a specialized cue for that just for this one area of social behavior, and it's also co-opted when she's not pregnant. But we thought maybe because it's so difficult for the pheromone receptor and the hormone receptor to be coordinately regulated in the sensory neurons that are going to transform a certain behavior, that maybe this was a feature in more of the sensory neurons. And so Nora Kableski in my lab decided to look at this. And she started by focusing on the molecule that was targeted to be silenced when the female's responding to the males, this PLC. And what we quickly realized was that even though the textbook said that PLC beta 2 was the primary signal transduction in these sensory neurons, that if we went ahead and took a PLC beta 2 mutant, so it did not have PLC beta 2 in these sensory neurons, and activated with this male cue, indeed, all the sensory response went away, as I showed you in that last slide. But if we did something more complex, like just allow the neurons to sense male urine, which has a lot of other odor cues that are important to the female, now you can see that much of the activity remains, only this small portion is gone. And if we presented the neurons with another special cue, that's the smell of cats, which is a predator of mice, and these sensory neurons are also not only involved in sensing social cues, but also predator cues, we find that all those remain. So whether she's in estrus or diestrus, she could still smell the cats, and she could smell a lot of the male cues too. And so this was surprising to us, because most other sensory systems that use GPCRs do so by varying the receptors so that they can detect a lot of different types of sensory information, but they all feed into a single kind of signaling motif. So whether it's taste or vision, they're all using one major signaling type of molecule, and that's what was thought for the vomeronasal organ. But in fact, that behavioral experiment with the PLC beta 2 knockout suggests that there's more than one type of sensory of signaling system present in these sensory neurons. And so we looked at RNA-seq and looked at the other PLC family members in these sensory neurons. So each neuron is expressing a different type of receptor to detect different cues, and that image is represented here. And if we just took them all together and looked at all the tissue, we found that PLC beta 2, the one that was important to sense the male cues, is just a very low number of reads, but that all the PLCs of the whole family seem to be present. These are all different genes, entirely different molecules. And if we just looked at the sensory neurons, none of the supporting cells or blood vessel, we find that PLC beta 2 is present, but we have evidence for at least 10 other PLCs that are strongly present just in these sensory neurons. And if we went ahead and deep sequenced individual cells, we could find that indeed there were cells expressing PLC beta 2, as I showed you before, that functional subset that's detecting the male cues. But individual neurons did not all express beta 2. Instead, they did express these other PLCs. And it wasn't as though they were all expressed in these other cells. Each cell is biased to express primarily one of these PLC variants. So what we have is evidence for many different types of signaling motifs in each of these kinds of individual signaling neurons. This is independent of the pheromone receptor, and that they're sort of mutually exclusive of each other. We've been able to functionally test, we focused on the PLC betas, and we've been able to functionally test, in addition to the PLC beta 2, which I showed you before. Beta 2 is present in just a very small number of cells. And when we go ahead and look at a marker for activation, this is very much like CFOS, which you may be familiar with, but CFOS doesn't work very well in the VNO, so we use another immediate early gene called EGR1. And if we look at EGR1 expression in the PLC wild type animals, there is some, and in the knockouts it goes away, and that's shown here, suggesting that it's, as I showed you before, necessary to functionally transform the detection of the pheromone into neural activation. And we could do the same thing with PLC beta 3. This is in a different subset of neurons. There's more of them. In the mutant, it's not complete loss of activity, because we have to find the cue that's going to activate just this subset of neurons. They're each responding to a different cue. So this cue activates primarily PLC beta 3, but a few more. But in the knockout, nevertheless, we find that when it's knocked out, that detection is dramatically decreased. And that indicates to us that PLC beta 3 is the primary signal transducer in those neurons. And we could do the same thing in PLC beta 4 cells. Here we have a different stimulus that activates these neurons. These are more predator responsive. And here they're not all PLC beta 4 detecting it, but primarily. So when we knock that out, most of the activity goes away again. And that suggests that in addition to PLC beta 2, beta 3 and beta 4 are also the primary signal transduction elements in their cells. And so here we have a big difference between all the other sensory systems that we know of, in that they all have this one primary signal transducer module. And here we so far have evidence for at least these 3 PLCs plus something else that's going on in the system. And it could be up to 16 different channels. We haven't been able to test them all yet. So why would this be? Oh my, I was supposed to, I have a different presentation that I didn't open. So what I'm going to do for time is say that let me see if I can go through this quickly. All the different PLC molecules are actually quite different from each other. They all have different capabilities. And while the PLC beta 2, as I showed you in the first story, when it was phosphorylated, it was silenced, completely downregulated. Some of the other PLCs they're all affected by phosphorylation, but in different ways. Some are upregulated, they're silent when they're not phosphorylated and they turn on, as these are here. And some are like beta 4, where they're silenced depending on which residue they're phosphorylated on. So all of these have the ability to be turned on and turned off, like we saw with the response to the female detecting the male predator odor. In addition, for this to be a system that's modulated, that's widely allowing modulation, we would expect that, like the PLC beta 2, which was specifically located in those cells that were responding to the male odor, that there would be some logic between the sensory neurons that are detecting a certain cue and the ability to have it modulated. And we have two other markers. So these are markers for receptors in the vomeronasal organ. This green marker is overlapping with a certain subset of receptors here, shown in these open histogram calculations. And the blue is shown in the hatched. And as you can see, the beta 2 is primarily in those cells that overlap with this green marker. And this guy over here, L1, is primarily in the cells that overlap with the blue marker. And that suggests that there is something that's determined about them so that they're likely to be segregated by the identity of the receptor. Of course, we have these guys in between here, H2 and L2 and E1, that are split evenly. And the green and the blue cells here have entirely different families of receptors that we think are doing different things. And so we're curious about this first marker that also these PLCs that are overlapping with both equally. Okay, in addition to be determined by their spatial position and the types of other markers that they overlap with, they're also determined by their abundance. So we find that they fall into 3 abundance classes, whether they're in a male or a female, a juvenile, or an adult. And so there's the more highly abundant PLCs, H2, B3, and L1, shown here with H2. There's these lowly but moderate ones, shown here with this H1. And then there's the ones that are very, very rare, and that's shown here with beta 2. And so we believe that not only are they expressed 1 per cell, suggesting that they have a function, we've functionally validated several of them, and that they're determined to be expressed either by their abundance levels or by their position in the vomeronasal organ. So they're not just randomly expressed in each neuron. In addition, we also find evidence for more hormone receptors in the vomeronasal sensory neurons. So the unusual non-canonical progesterone receptor that I showed you earlier is called PGRMC1, but there's several other types of receptors that are expressed on these neurons that could also modulate the response to other social cues. And then we find other interesting motifs that respond to stress or hunger and satiety. All of these receptors are present in the sensory neurons, giving it the capacity to respond to a wide variety of internal state hormones. And so we wanted to ask whether there was widespread changes in signaling depending on the internal state of the animal. And to do this, it's very difficult for us to find a ligand that activates a particular subset of neurons and figure out an internal state that would be modulated by it and read it out by a behavior. And so to go one by one, like we did with the sensing of the male cues, is very difficult. So instead, we did this more broadly, like we did with looking at the signaling molecules. And we imagined that if we take an individual animal and there's no modulation, the response to a single type of odor cue should be the same in every individual that we look at. And that's what we assume are passive sensation. But if we take different types of individuals that have a different hormonal complexity, juvenile, adult males and females in estrus and diestrus, as well as pregnant females, each of them will have a different hormonal profile and perhaps cause modulation of the system. And so we could do this by using a marker that allows us to pull down the transcripts of the activated neurons. And by that, we were able to do qPCR on those markers. And by the qPCR, we looked at juveniles compared to males, compared to females in diestrus. And as you can see, they all naturally cluster and the responses are different. These are transcripts from just the PLCs. So we find about 7 different kinds of PLCs that are activated from this odor that we gave each of these animals, which the odor was all the same, but the response of the neurons is different. So whether you're juvenile, male, or female, you're all turning on neurons that have H2 and L1, but only in juveniles do you turn on these that have beta 3, D4, XD2, and only in males do you turn on sensory neurons that have H1 and L2. These neurons are silenced, these neurons are activated. And that's shown here by immunofluorescence, and this was the qPCR. And this not only corresponds to the PLCs, which are changing, but to validate it, we could also look at the pheromone receptors themselves, which shows it's a different sensation. And here they're in a different order, I apologize, but this is the adult male, which is shown here. This is the juvenile, which is shown here. And this is the diestrous female, which is shown here. And you can see that the patterns of receptors activated are different. So by a sensory point of view, we would say that each of these animals is having a different perception, a different response. And by our molecular experiments, we're postulating that perhaps different neurons are being turned on and off due to the different hormones in these individuals, the hormone receptors on these sensory neurons, and the modulation on these signaling elements. So what we found is that instead of just a passive selection where everybody's sensing the same thing, giving the same information to the brain, and the brain is deciding how it should act depending on the internal state of the animals, we believe that this could be indication of a personalized sensation where the nose itself is using this if certain hormone then either turn on or turn off the sensory neuron. And so each individual has a different representation in the environment, and that's underlying the different behaviors that we're seeing at these different stages. Now, this was really surprising to us because we entered this project hoping to study the brain, and we ended up studying the nose. And our view of the nose was that, this is a very simplistic view, which we can talk about later, but just a passive feed-forward view where first you sense something and sensation drives behavior. And then once you sense it, you need to decide whether you smelled it before and what's out there and what your response to it was, whether it was good for you or bad for you. You have to decide on your internal state. If you're hungry and you smell food, you would want to approach it. If you're satiated, you would ignore it, and other internal state cues play a role. And then you also need to know what the other sensations are that the other sensory streams are giving you. Do you smell food, but you see a cat? Then you shouldn't stop to eat, right? So all of these decisions are sort of fed in once you smell something. There's all this decision making before behavior can occur. And what I just showed you was that this sort of feed forward from very simple sensory processing to more complex decision making isn't necessarily linear and as I just described. What I just showed you was that the response of the body already is telling the nose outside of the brain to compute a different kind of information. And so we're very interested in understanding this modulatory effect of the olfactory system, just for a practical point of view, that if we can then understand it and clamp it and control it so that every individual has the same sensory input, then we'll be able to study the brain a little bit more reliably. So that's the first thing. And the second thing is this understanding that we don't really know where in the brain decisions are being made, if the nose can be one of those places that's making a decision. And we want to understand how this information is being transformed from sensation to behavior. And for social behavior, we don't really understand the information flow. And part of that is because many people in our field just find one place, either in the hypothalamus or in the amygdala, and they focus on that to study social behavior. And we feel like that's a little bit like the blind man sensing an elephant, right? Where one person sees a tree or a wall or a snake. And really what you want to see is the elephant. And in the brain, we really want to see the elephant, not just the whole thing as a system, not just the individual players. And so several years ago, we wished to find an entire sensory to motor circuit, if it exists, if it's even possible, so that we could then focus on just those places in the mouse brain and study what's happening during natural behavior. And this has been really hard to do because most social behaviors require a lot of sensory streams, not just olfaction, but vision and auditory cues. And so there's a lot of streams coming in, and then there's a lot of motor groups going out. You can see these behaviors are requiring tens or hundreds of motor groups, and they're used in different orders that vary on millisecond timescales. And so really getting that whole stream of sensory to motor has been very, very impossible. Until I had a graduate student, Jason Keller, in the lab, who suggested that we use this very simple behavior and convinced me that it's a nice model and a nice social behavior. So this is a behavior that's social with just one animal. And as many of you may know, male mice, when they smell the odor of female, her pheromones elicit him to be in courtship mode. So he wants to advertise to her. So here's a camera on top of the mouse's behavior, and underneath, you can see that once he starts smelling the female urine, he started urinating himself. And if you can hear these little whistles that he's making, I'm not sure how well this is playing, but he was making a buzzing, whistling noise. And those are ultrasonic vocalizations. These are at a frequency range that our human ear can't hear, but we slowed it down so you can hear it. So it's sort of a private communication. He smells the female's pheromones. He emits his own pheromones to attract her. And he starts calling in his private language so that perhaps he can find her and they can mate. So this is a really robust behavior that's occurring without the presence of it's just one sensory stream. So it's very easy for us to control. Here's just a quantification of that behavior. So it's a 2-minute assay, and you can see for the first 2 minutes with the control odor, there was very little spontaneous urination, only when there was a physiological need. And then when we added the urine, as you saw in the video, these 12 mice largely began to mark those urine spots. Each tick is a spot quite rapidly. And this is the calling. And they do it interleaved. They call and they sing, as you saw in the video, evoked by the female odor. So we also think this is a voluntary, not just reflexive peeing, because they're not doing it prior to the sensory cue. And on the sensory cue, like if you see the sign for a restroom nearby, then you will start, you will begin the process of urination. These guys are voluntarily beginning the process. And this sensory circuit, like those more complex social behaviors, is also modulated by some interesting computations in the brain. So it's developmentally regulated. Juveniles will not call and mark to the female urine, only adults. And females do the behavior differently than males. So it's sexually dimorphic. It depends on experience. So subordinate animals do the whole repertoire of behaviors entirely different than dominant males. And it's modulated by internal state changes, such as whether the animal's thirsty or sleepy or stressed. All of that will change whether how the male performs this behavior. So it has all the interesting coding elements, but it doesn't, it's not as complex. It's very simple. And the other advantage is that it is a very simple behavior on the motor groups. So for the calling, you just need pressure from the diaphragm and the larynx. So that's very simple muscle groups. And for the urination, you just need the bladder to contract and the sphincter to relax. And once the sphincter relaxes, then urination can begin. So this is our target voluntary muscle group here. And both the diaphragm and the sphincter are the target muscle groups for the calling. And so by starting with these known muscle groups, we could trace backwards into the brain and begin to find the circuits. And it was previously known that there's a region in the brainstem that controls urination, but nothing else in the brain was understood about how voluntary urination occurs. Most of the study has been from the spinal cord to the motor elements. And so when we first started this project, Jason Keller in the lab discovered a new subset of neurons that are expressing estrogen receptor in this region of the brain that had previously been known to control urination. But the understanding of how the neurons controlled it was unclear. These neurons project to the spinal cord, as shown here in this panel from the green neurons here, in a region across this DGC that is known to connect to the sphincter and allow for voluntary urination. These other red neurons, as well as the green neurons, these red neurons that were previously studied by Bernardo Sabatini's group and others, project only to the bladder regions, and they're known to squeeze the bladder. But if you squeeze the bladder and you don't relax the sphincter, all that becomes is that you're uncomfortable. It doesn't cause voluntary urination. So we decided to focus on these green neurons. And so Jason was able to perform fiber photometry put GCamp in just the subset of green neurons and look at their activity during urination. So the yellow ticks here are the urination events, and this is a raw trace from the fiber photometry, and you can see the correlation of neural activity is quite nice. And if he goes ahead and puts channel rhodopsin in these neurons and then stimulates with light at increasing frequencies, again, we have a camera above and below. As he moves to 5 hertz here, you can see immediately he gets a urination event. And as we stimulate stronger and stronger, the urination comes out more robustly and rapidly. So you can see here that was 10 hertz, and it will increase. So these neurons are sufficient to generate urination. If we do the same trick with the neighboring neurons, no urine is released. And we can also monitor this social behavior when the animal's anesthetized, which is kind of nice to understand how the neurons are working. And so here in the, we can do something called cystometry, where we fill the bladder up with saline, and we can stimulate those green neurons in the brain and record the effects. And so here, this is the catheter to add the urine. And as the light came on, you can see that the urine starts to come out. And if we go ahead and run it again, you can see that here we've looked at the bladder pressure, which increases, and the sphincter is bursting. It's like a peristaltic pump. So it bursts and relaxes, and it's during that relaxation phase that the animal is urinating. And then we could also see if it was sufficient. And so this time the camera is just on top. We're going to start with the lights on to turn off these green neurons. We add the female odor. And then we find, very interestingly, that the mouse itself is still interested in the female odor. So he knows that it's there. He went over to it directly, not to the corner, and he sniffs it. There's a little bit of urine that comes out, but the response isn't at all like you saw in the first video. And then if Jason turns off the light, you can see already there's a halo beneath him where he started to urinate right away. And then he goes right into the marking behavior right away. And so this is quantified here. This is lights on. This is when the odor was added. You can see all the animals are generally sniffing the odor. So that suggests that what I showed you in the video is reliable. Their sensation is still there. And most of them do this peeing right after lights off, suggesting that the system was primed. If we go ahead and add the light without the odor, so the animal's still expressing the arch T, we turn on the light, we don't get this rebound effect. So it really suggests that the system is primed, the information is flowing into the brain, but it needs these green cells to be able to urinate. So, because we found these new neurons here in the brainstem that are projecting to the spinal cord and then to the muscle and controlling necessary and sufficient for this response to the female cues, this voluntary urination. They're a very small number of neurons, and so we've been able to use those as starter cells and really start to be able to find a basic skeletal circuit from the nose all the way to the muscle. These regions here we have not interrogated or validated yet. They're just our closest way to get from these sensory neurons to these centers that we now know are important. So maybe at this point that they connect to 20 other places in the brain and we can't get clearly back to the nose. But as of now, we're hopeful that we'll be able to just walk through it. And then this will give us sort of the whole elephant. And we'll be able to use fiber photometry across the whole system to study the events and features and how the behavior is taking place. In the meantime, as we were interrogating this region, the LPOA, instead of it generating urination, it evoked the calling behavior. And so that's shown here. If we go ahead and drive channel in these neurons, whether the animal is male or female, they start to do these ultrasonic vocalizations quite rapidly and very robustly. They'll call until they're hoarse. Oh, and so here, actually, that's shown here. Sorry. So if this video is playing, you'll hear the noise again, and this is a sonogram of it. So the light turns on the behavior quite robustly. And fiber photometry during natural behavior also suggests that these neurons are naturally active during regular behavior. Just like with the urination, we could see if it's sufficient by blocking the activity of the neurons. Oops, sorry. Let's see here. Let's see. So here we're going to start with lights off. So the neurons are active, and he's interacting with this female here. This is natural calling. We're not turning it on. And then Jiggy is going to turn it on to block the neurons in the LPOA. So the male stops calling suddenly. And just like with the urination, he still recognizes that the female's there. He hasn't lost his sensation or his motivation to interact with her. So we believe that it's also, we're catching it at the LPOA at this transformation of the information. Excuse me. We were able to look at the types of calls. So a lot of people think of USB calls as a language. Mice make lots of different syllables, slightly different noises. And if we look at the repertoire of calls between our artificially evoked, those evoked by channel that I just showed you, compared to natural calling repertoires, when the male is either interacting with other males, pups making their spontaneous cues, or when the male is interacting with females, we find that the artificial syllables are exactly broad and represent about the same complexity as the natural cues. And that if we were to align them, they're more similar to when a male is detecting a female. But we don't think that's important. What we think is interesting is that it's not just stuttering or making the same syllable over and over again. It's able to make the whole repertoire of natural cues by stimulating LPOA. The next thing is that there was more known about the calling circuit than about the urination circuit. So motor centers, perioctaedicular gray are both feed into this region in the brainstem called the nucleus ambiguus. And so we asked, is this new region of the brain also, is it feeding in directly to the ambiguous, or is it interacting with other known parts of the circuit? And we found that it sends direct functional projections to the perioctaedicular gray, and from there it's eliciting the calling. So it's plugging into the known calling circuits. And so that diagram is here. These are our new neurons that we found to generate calling. And the output neurons that have been previously described by Fan Wang and Rich Mooney are excitatory neurons in the perioctaedicular gray that then drive the action in the larynx and USV calling. And our neurons ended up being inhibitory neurons. They're all these inhibitory long-range projection neurons. And so we can show that both functionally here by doing fiber photometry on them, as well as we identified a cloud of inhibitory neurons that surround these output excitatory cells. And if we drive channel in those neurons during naturally evoked calling, now these ticks here are representing the USV calls, we find that when we turn on these cells, the calling ceases immediately. And so that suggests that this is the circuit. It's a disynaptic disinhibition that's allowing these cells to turn on. Now, that sounds a little wonky to me, disynaptic disinhibition, why do we care? So what I assume is that it's going to provide features that are important for the behavior that a pure excitatory signal couldn't generate. And so we decided to look at this, and I'll try and be quick. We decided to look at this not in terms of the mouse generating a language, because there's no evidence in our reading of the literature that USVs are structured like a language. Instead, we think it's more like non-language verbal communication that your many animals do, and humans do as well. A baby cries or laughs, you giggle, you scream. There's a lot of important information in there, but it is there's no language at all. And so we decided we were wondering whether that's what the USVs act as in the mouse. And inspiration also came from Jiggy's cats. So if you can hear these videos, there's different kinds of cat noises. That's cat food. These animals are excited to be fed. And this is the cat in an unpleasant situation for it. So it was making different kinds of noises, and nobody's really looked at, and the difference between those noises are the scaling, either from short and quiet to long and loud. And with human noises too, whether you laugh or cry, you know that it's the scaling that contains the social, the emotional information. So we ask, do mice USVs scale under the natural context? And so if we take a male and we record his natural vocalizations, whether he's interacting with a female, female odor, anesthetized female, male odor, or an anesthetized male, we can find that not only the amount of USVs scales, but the amplitude. They're louder when they're interacting with an awake female or female odor, and they become quieter, as shown in these lighter colors, when the females has all the same sensation, but she's just anesthetized. And also the bouts, sort of the sentences, the strings of syllables that are strung together. If we break them up when they have a 2-second pause, so they still go, but they're taking time to get more breath. They take a break, and during those times we could shuffle them around and quantify them, and we can see that when they also scale by this length. So they're longer when it's responding to these an awake female than when he's responding to an anesthetized female. And she really is emitting most of the same cues except the visual cues of her movements. And so we really do suggest that there's these motivational differences, like a human laughing or crying. And so here we can now ask, is this feature of scaling present in this part of the calling circuit? And we find that when we just stimulate at very low frequencies in this region here, we find that the calling comes on very loud, and also it's very long. So this was the 5 seconds was as long as the light is on, this 5 hertz or 10 hertz, and it's really filling up most of this time here. But if we stimulate here in these new neurons, we find that it's scalable. So even down at 5 hertz, if you compare these two, it's generating very little noise, and they're relatively shorter, and they get progressively longer, the bouts, as we scale up. So the scaling feature is emerging here by this arm. And the other feature is the persistence. So this is now not just the bouts, these 2-second pauses remain. This is the whole conversation. And you can see that when you stimulate here, as I showed you in the last slide, the calling is locked to the light. It goes on and off right with the light. But when we stimulate here, it comes on right with the light. But after the light goes off, and you may have seen this in the earlier video, the male will continue to call. So he's just excited to maybe see her, and he just won't stop. And we found that feature emerges here. This is a slice recording of the stimulating axons from the LPOA that have channel to turn it on. And when we drive the channel here, we can see, remember, these are inhibitory neurons, so it's turning off these cells with the light. But they don't, when the light goes off, these cells are not recovering synchronously. They don't recover right when the lights come off. They are, some come right back on, but others are delayed here for several seconds. And that's coincident with this variable and delayed calling behavior that we're seeing. And so we think that this circuit, this part of the circuit, by having this disynaptic disinhibition, is giving us these features of scalability and persistence. Okay, and so what we've done is we're looking for this elephant, this primary circuit for 2 different behaviors, calling and marking. And once we have it, we like to use molecular biology, genetics, and cellular activity monitoring, such as neural pixels or calcium imaging, to be able to understand just the information flow. Is it simply this feed-forward thing that we imagine, or is it dynamic? And if it's dynamic, where? And how does that generate to the features that we see in the output behavior. For the events, I showed you USV calling and urination events, but the animals were also sniffing and walking. There's other motor behaviors. We like to map how all of those map into the system so that you have sort of this active sensation with the sniffing as well. And then all those interesting features, such as reward or arousal or valence, are all encoded somewhere. Probably not directly in this circuit, but there's going to be edges, just like with internal state. Where does stress feed in? Is it all just in the nose and the nose is regulating this? That would be silly. It's also got to be regulated in other places. And so where are those edges and how do the patterns of activity change as internal state changes? And so overall, we're hoping that this will allow us a more complete picture of the computations of social behavior. So all of the work that I described in the first project was done by Sandeepa Day and is now being done by Nora Kobleski. And Jason Keller did the urination work, the urine marking work, and Jingyi Chin as focused on the USB calling. And we did it in collaboration with these wonderful people across the US and the world. So thank you very much for your attention. I'm happy to take questions and hopefully next time we can meet in person.
Thank you, Dr. Stowers. It's for a very nice talk. Now we'll take few questions. Shabnam, do you want to come live and ask the question? Shabnam Chande. Okay, so let's see if I.
Hello, am I audible?
Yeah. Yeah.
Thank you, Dr. Lisa, for that amazing talk. I was actually wondering that when we block the signal to the vomeronasal sensory neurons during the elevated levels of progesterone, does that also mean that other cues that are sensed by those sensory neurons also get shut off or are detected not as efficiently by the female during her distress phase? You were talking about cat pheromones. I saw that during the presentation. So how is that regulated since the signaling cascade is blocked by the pheromone, by the progesterone binding to the receptor?
Yeah, this is a great question. It's one that concerned us very much when we first saw the silencing, because we were worried that she wouldn't have any sensory cues while she was in the diestrous state. And it's a little bit confusing, but those sensory neurons are all different, just like different if you're more comfortable with T cells and how each T cell is able to respond to a different immunogen in the immune system. The sensory neurons are each able to respond to one or a small number of odor cues. So those that were affected or silenced by the progesterone were just those that were expressing PLC beta 2. That was a very small number of neurons, and they're just associated with a very small number of pheromone receptors. And all the other neurons were still active and able to sense things like cats and the other male cues as well. So she's not completely blind. And our last experiment suggests that it's really dynamic, depending on the internal state of the animal, or this is our hypothesis at this point, that different sensory neurons are being turned on or off based on the needs of the animal, so the animal can focus. We're imagining that we still don't think that the female or any individual, a hungry juvenile or a male, stressed male, should be blind to some cues. So we're imagining that this particular sensory system is the one that's modulated, whereas something like the main olfactory epithelium or obviously the visual system is going to provide a constant stream of what's really out there. And that maybe the function of the system is just to give the animal the attention to it or something, the extra emphasis. So without it, the animal just, it still knows the cue is out there, but it's not so important to it. We're still not sure.
All right, thank you. That does clear my question.
Okay, so there's next question by Itakura. Do you want to come live and ask the question? Okay, so I can read the question. So he asks, regarding knockout of PLC genes, I wonder why male urine induced EGR1 in VNO less than the steroid pheromone. I think male urine contains much more components than the steroid pheromone.
You're exactly right. The steroids were actually a blend of just a couple of them, but it was a very limited number. And male urine should have several hundred odor cues in it. The vomeronasal organ, so this is that specialized olfactory epithelium. There's also a main olfactory epithelium, as I was referring to sort of in the last question, that that can detect all of the components in the male urine. And the vomeronasal organ just gates on a few cues. So the fact that we're getting more activity with sulfated steroids, it just means that that cue is being represented more in this organ. Even in the main olfactory epithelium, though, only about, even though there's hundreds of ligands and there's a combinatorial coding there where many receptors can detect many different kinds of cues, you only see about 10% of the neurons ever activated at one time. We don't understand how that silencing is occurring. There's beginning to be new studies from the Firestein lab and others that suggest that there's competitive antagonism in odor blends that's allowing for this sensory filter. Because otherwise, when you have something like white wine or coffee, where there's many hundreds of volatile cues, you would just get white noise if every receptor was activated. So there is some mystery on the transformation of natural odor scenes. And our male urine in this case is one of those.
Okay. So we'll take next question from Arun Kumar. So do you want to come live? Arun Kumar. So I'll ask the question. Even though it is just an odor and there is no other actual animal inside the cage, why mouse would still avoid the area around the odor coming from?
Right. We find this most dramatically when we use predator odors as well. And during these short assays, so these are shorter assays. The one that I showed you in the slide that I think he's referring to was a longer one. It was about 20 minutes. In a shorter assay, when they find something that is aversive or not interesting, right. So how would they avoid it? It does suggest in that data that it was avoiding it for some reason. It was noxious, and we don't understand why the female would avoid a male odor. But that's something with their social behavior. But generally, the root of the question is, it's just an odor. It should be able to see that there's nothing scary there. And indeed, that's the case. So if we use a predator odor, a snake or an owl pellet, the mice are terrified. Their hormone levels go through the roof, even though our lab animals have never, ever interacted with a predator before. So there's a very strong detection through the vomeronasal organ that makes them very scared, but only for about 15 minutes. And then they sort of recover and they start to investigate. And if you let them go for 30 minutes or an hour, they'll start flipping around the odor cue and playing with it because they're bored. And so exactly as you said, they are able to learn and respond quite quickly that it's not scary. And we think that's important because they're the low predator, right? So if they're out in a field and they run through a territory where a fox or a coyote was before, and they just freeze, if they stay frozen forever, they're gonna die. That's not a good way to get food and escape other predators. And so we think that this system of just the odor cues is very robust, but it's just one piece of the puzzle. And so it gives them a warning, and then they can cautiously move forward, but not be frozen forever.
Okay. So the next question is from Ankush. Ankush, are you coming live to ask the question?
Yeah, hi. Thank you for the wonderful talk. I wanted to ask, is there neuroplasticity in the nose itself, and could that make us more skilled at recognizing certain specific smells?
Yeah, I don't study that directly, but there's new data coming out that suggests so that our human nose is always changing. Every 60 to 90 days, it's one of the neuronal centers that has a very strong neurogenesis, and your whole epithelium turns over every 60 to 90 days. The way that it turns over can be biased. There's some experiments that suggest that if you have especially young exposure when you're young, where there's even more exuberant turnover or birth of neurons, that you can bias the representation. So the size of the first relay in the brain to one odor will get larger, and the number of neurons will get larger. Functionally, it's not clear whether that makes a very big difference. The percept is a little stronger, but it's not. So and it occurs over very slow timescales, so months. In the mouse where we've been able to interrogate it, months in a mouse's life, they don't live that long in the wild. So, so really trying to, it's not something where there's just experience and then the whole system will change over rapidly, sort of in a timescale that makes sense in a week or so. We haven't really seen much of that. I think, moreover, the system is good at just giving a lot of diversity so that it can detect a lot of different kinds of chemicals. And then the brain just learns, and that's where the brain comes in, to associate the meaning of what this new thing is with whatever was emitting it as the animal develops. So rather than biasing the sensation, I think it's allowing a system that allows for broad sensation.
Thank you. Yeah. So the reason I was asking this is, like you showed in your data, how certain at certain points of the estrous cycle, they might still perceive it by the nose itself. I was thinking, could certain life events perhaps make certain changes in the nose, which would make them, allow them to get such perceptive cues?
Yeah, as of now, we don't have anything that's changing on those types of timescales that would change within estrus and diestrus or hunger and satiation. But this sort of modulation could provide that sort of thing that you're looking for. So we're still looking, and it's still a work in progress. I'm not sure if it'll all hold up, but we're excited to look at that question. Thanks.
Thank you.
So the next question is from Srish, and Srish, do you want to come live and ask the question?
Yeah, sure. Thank you so much for the talk, Professor Stowers. My question is a little quantitative. So, at what concentration of the pheromone is this pre-mating behavior elicited? Because, as you pointed out, in the wild, there would be a cocktail of odors as well as other sensory cues. So, I was just wondering if the pheromones in different concentrations or in combination with other odors would be able to elicit the same intensity of response, or how would it differ?
Yeah, this is a question that we're just trying to gear up to start to begin to answer now. It's been very hard because generally the transformation, even when we provide an odor cue, either, as we did the urine in this countermarking arena that we had, which was just adding it, that was natural concentrations. That was just pure urine. Or if we deliver it either through the air or through other means, it's going to get dilute in the air, in the vapor. And then as it enters the nose, it's going to dilute more. And then there's mucus that it has to dissolve in. So, there's, nanomolar concentrations are effective in the main olfactory epithelium for generating really robust responses. And we can determine the concentration, using our perfusion mechanisms on sensory neurons, but without the other environment, we don't even know if those are natural concentrations, and nobody's been able to record the concentration of ligands that are transferred between individuals. So we have a new collaboration with someone here at Scripps that has now been able to label the pheromones, and we're watching them being transferred from animal to animal and doing mass spec then to see what the effective concentrations are. And so we can use neural recordings to look at neural activities and look at the effective amount just to turn on the cells, and then look at how that correlates with the effective amount to generate behavior as well. So when they're in a mixture, right now, all we can do is do these artificial blends. If we put a male and a female together, and a female with two males, we find that they'll fight first and then mate. So there is some sort of a hierarchy, but and whether the animal, when he's in his fight mode, can he sense the female? We don't know. So this is an excellent question that honestly the field, even with regular odors, how can you smell orange and coffee at the same time? It doesn't just become a crazy, your breakfast isn't one big blend. You can smell the different components. And those are generally bounded by sniffs, and that's packaging the information together. But it's all a bit of a mystery.
Yeah, thank you so much. That mass spec incorporation to your research seems really cool. Thank you.
So, Dr. Stowers, I had a question. See, when you present a cue like a pheromone to the animal, so after a point of time, the animal will become habituated to a particular concentration. So, does there is a kind of neuronal plasticity or memory, like a memory-related plasticity in this, that the animal get habituated to a specific amount of pheromone, and does it changes with the age also?
Yeah, that's also a good question, because the animal responds differently with age to these to pheromones, and some of that has to do with experience and development, but some could be the nature of the experience. It could just be the exposure. We don't know. The short answer is we don't know. We do know early work suggested that unlike other sensory systems that have a very strong habituation at the sensory molecular level. So an odor, if you walk into a room where you smell bread, you smell it very strongly immediately, but several minutes later, the odor isn't so strong. And that's because the receptors themselves are being downregulated and recycled and turned off by arrestants. In the vomeronasal system, at first, we didn't think that was happening at all. We could give an animal an odor and the response would be persistent, but it does seem like some odors do cause habituation, and we don't understand the molecular differences between these and the significance to behavior. And so I think that's another interesting focus to try and understand the evolutionary pressures that drive those neurons to adapt versus those that aren't adapting, and how experience can drive behavior overall. Thanks.
Thank you, Dr. Stowers. That was a very nice talk. And now I'll hand over to Pranjal.
Hello, so I am Pranjal, current president of Project Encephalon. Thank you, Dr. Stowers. It was a very exciting talk. Thank you so much for being with us. At the end of each talk, I tend to acknowledge some members of my team without whom organizing Cajal Week would have been impossible. Today, I'm acknowledging the work of Navodita, Jay, Ritika, Svikrut for providing extraordinary support during the event. I am also grateful to Scientists Inc. for sponsoring the event. Next talk will be by Dr. Leah on 6th May, Thursday at 6 p.m. Indian Standard Time, followed by interactive social hour. So please do attend that. I will be posting the links for that in the chat box right now. Thank you so much, everyone, for attending the talk. See you on Thursday. Bye.